Hello, everyone, and welcome to this week's NeuroTools Webinar. Today, I have the pleasure of introducing Michael Feolo as he shows us the NCBI's database of genotypes and phenotypes, otherwise known as dbGAP or dbGAP. Mr. Feolo is a staff scientist at the National Institute of Health's National Center for Biotechnology Information, and since 2007, he has been the team lead for the database of genotypes and phenotypes, dbGAP. The dbGAP is a database designed to allow researchers public access to questionnaires, protocols, methods, phenotypes, genotypes, and the results of association and linkage analysis, in whole genome case control and longitudinal studies of heritable disease. Since 2005, Michael has also been a team lead for the dbMHC database, a web interface and permanent public archive of genotype, phenotype, typing kit, and clinical outcome data concerning the variations in the HLA genes located in a region on human chromosome 6 known as the Major Histocompatibility Complex, MHC. Without further ado, I'd like to hand the microphone over to Mr. Feolo. And Mike, if there's anything you'd like to add, please feel free. But I'd just like to take a moment to thank you for joining us today. And we're all looking forward to this webinar.
Thank you, Devin. And thank you for inviting me. Hopefully everybody can see my screen. I'm going to talk today about a database of genotypes and phenotypes at NCBI. The outline of this talk is, I'll start with just describing what dbGAP is, and then I'm going to go into how to get your study registered if you would like to submit to dbGAP, briefly how to format the data for submission, and then how to upload data, and then I'll briefly touch on how to get access to data at the end. Just to orientate you to what dbGAP is and where we're located, it's part of the NIH in the National Library of Medicine at NCBI. And NCBI is probably most widely known because of PubMed and PubMed Central. It's a literature database for all kinds of journal articles, and it covers anywhere from the genome information through variation, phenotype, all the way to medicine. The genome browser is another common tool that people know NCBI for, but there's a lot of other tools at NCBI. And if we go back to 2000, when I first started working at NCBI, we had resources, and many of you may be familiar with GenBank or the Entree Gene or Gene database at NCBI, dbSNP, which is a database of single nucleotide polymorphisms or small variations on the genome, and then GEO, which is an expression array database, and then RefSeq, which is a curated version of the GenBank sequences, and of course OMIM, which is a phenotype database, was based on literature from John Hopkins Group, and then it was done online through NCBI. And I started working on dbMHC when I first came to NCBI, and this is about in 2000. And the trace archive was built, and this was for Sanger sequencing information that would be deposited from sequence. And then in 2007, we started a new database that captured information from largely at that time GWAS studies, and we have a tool called Figini that shows the results of the GWAS studies. And then in the intervening the years after that, a sequence RRID archive was developed at NCBI to house next-gen sequencing. MedGen, which is a resource devoted to medical genetics nomenclatures, and then the GTR, which is the Gene Test Registry, which really goes into the domain of medicine at that point. That's just to orientate you where dbGAP is at NCBI. Talking specifically about dbGAP, the purpose of dbGAP is it's a permanent archive. We provide public accessions for data that is from any kind of research on that involves the collection of phenotypes on humans and molecular data from humans. And the data objects are versioned. There's two modes for dbGAP. There's public pages that has summary and meta-level data, and then there's a controlled access for individual level data. And dbGAP, and this is a figure from our Nature Genetics article in 2007, the white paper for dbGAP and how it was organized. And this data model actually holds true still today. But the way dbGAP is organized is there's a top-level data object called a study, and that's a collection of data that was collected together and analyzed together as part of usually from an NIH grant, but it can be from non-NIH funding also. The study would include phenotypic data, and we've modeled phenotypic data in dbGAP in a very general sense so that we can take a large number of different types of studies, so that we're not trying to cram a square peg into a round hole. We modeled it very simply, where we would collect the patient's phenotypes as a rectangular table, essentially, where the rows are people and the columns are attributes of those people, or phenotypic attributes or exposure variables on a person. And then we accession each table of data that we get, each phenotypic table we get, with a PHT number. The study itself is labeled with a PHS number. We also accession and version each column of data. So here we have a column of data that's the smoker status, and it gets its own accession. There's another column of data for current age, and that gets an accession, and so on. We have accessions for genotype arrays, large genotype arrays where person and variation are the axes, and the cells are the genotypes. And we can have a number of associated data like quality scores or allele intensity. We can also package a set of sequence data for the same individuals, or expression data, or other types of omics data. So that's just to kind of give you an idea of what types of data are in dbGAP. And this is just a written description of that. We have phenotype data, which is demographic, clinical, biomarker, exposure variables. It would also include any images. Study documents, which would be questionnaires and protocols, data collection instruments, where they collected information on the patients or the study participants, and then molecular data, which can be essentially any kind of omics data, but originally dbGAP was focused on array-based SNP data and copy number variants. We also have a large number of imputed genotypes, sequence-derived genotypes, and this takes in the form of BCF and MAF files, which are primarily tumor normal comparisons. And then we have a number of studies that have next-gen sequencing, either whole genome or whole exome. And then this would also include RNA-seq or methylation or expression data using next-gen technologies. And then the last kind of broad category of data is the association analysis results, which are the p-values that result when you analyze the phenotypes to the genotypes. Talking more about the phenotype data files, as I said, we modeled this as rectangular files accessioned with the PHT number, where the subjects or samples are rows and the variables are columns. These could be longitudinal, and in this case, we would have the key of the table to be the subject ID and the time point for each measured variable, or if needed, time point in place. With longitudinal type data, we have limited ability to show summaries on the public pages, but the full data sets can be available for controlled access download. And then we match a data dictionary to each data table that we receive that would have the data types, the coded values. These would be the code books in some studies. I wanted to give you a sense of the data that's available in dbGAP. And to do that, I'm going to actually bring up the public website, which is unadvisable in a live demo like this, but I'm going to try it anyway. So I clicked on that link, and it brings up our summary statistics page. And here I can show the growth of studies from 2007 when we started up until today. There's 951 studies available. In those, we have about 5,800 phenotype data sets. These are the tables I described earlier. And across those tables, there's 260,000 variables. And all said and told, there's about 1.6 million study participants represented in dbGAP. These are unique individuals. And from those individuals, there's about 1.8 million molecular samples that were drawn from them and tested on some assay. We also have a number of study documents, although this is not a large number. These data can be very valuable and are somewhat unique to dbGAP in that we link the study documents, data collection instruments back to the variables, and this is an indicator of how that data was collected, and so you can see the actual forms that were used in the study. If we go to the molecular data distribution, we see that about 1.3 million of the samples have array genotypes. Now, each number in this 1.3 is an array of hundreds of thousands of SNPs. And so the numbers of genotypes in dbGAP are actually in the trillions with a T. And this just summarizes genotype assays, and then these are the counts of genomic sequence assays. So these are whole exome sequencing, about 150,000, and then whole genome sequencing across the whole genome is about 54,000 today. And then we have other types that are less numerous, but important to some research, of expression arrays, epigenomic data. We have also tumor normal comparisons and different somatic mutation data, and then also microbiome data where they're looking for the bugs in human samples taken from humans. If we look at the use of dbGAP across the world, we see that it's most widely used in the United States, but we have quite a few users in Europe and in China and even in Australia. The number of data requests also follows that same trend, although the number of users doesn't always represent the same number of requests. Some users use a lot of different data sets. So this just summarizes that, and then you can see the growth of the requests over the 10-year period of dbGAP's existence. So now we're at about 41,000 requests so far for different data sets in dbGAP. So I'll just close this and continue on with the presentation. I did want to say that dbGAP is governed by NIH policy, and specifically at the moment, the genomic data sharing policy. And if you want to learn more about that, you can click on this link. Devin, I'm wondering, are the slides going to be provided to the people participating today?
Hi, Mike. Yes, we could arrange that. And I also want to point out that we are recording this, and it will be on YouTube just a few hours after the live presentation is completed. We can also provide access to the slides themselves if you'd like. You and I can coordinate on putting them on a Google Drive or something like that if you would like that as well.
Okay, yeah, some of these URLs are long, so it would be nice to have it. Okay.
Sure, absolutely. Yep.
Yeah, okay, so the genomic data sharing policy is available through the NIH website using this link, and you can read about it. The policy itself is well, you can read it. And there's other information on guidance to people that want to submit, or how to get access. So I'll continue down. I want to now transition to the phase of this talk on how to submit for those of you who might be watching this that are interested in submitting data to dbGAP. The place to start is to go to the dbGAP homepage. The URL is here, and on that homepage there's a link called How to Submit. And when you go there, you see this flow chart. And the flow chart kind of talks you through. The first question we ask is, well, let me describe this flow chart a little better. This flow chart has 3 horizontal swim lanes where the principal investigator, genomic program administrator, this is an NIH official who would register your study, and then dbGAP. And then going across, we see that there's 3 phases. There's a study registration phase, a data submission phase, and then study release phase. And so if you were interested in submitting data to dbGAP, you need to first start your registration. And then the first question is the NIH, is the study NIH funded? And if it is yes, then you need to contact your program genomic program administrator. If you click on that link, you'll get a list by institute at NIH and the contact for that genomic program administrator, and you can contact them directly and say, I'm ready to share my data for my NIH study. It turns out that if you're not funded through NIH, the process is somewhat similar, but you end up contacting a genomic program administrator, but you have to figure out which one is most appropriate. But if you follow the NIH guidance, you'll kind of get back to this list. And then at that point, this genomic program administrator can start the registration. And the main thing for this registration is there's some paperwork that needs to come from the submitting institute, and that's the institutional certification. So that's really something you want to look at first and start getting that filled out. And that this institutional certification basically says that your institute agrees to you sharing the data through a controlled access system and dbGAP. And once the institutional certification and some other study level information is entered into the registration system, it can be completed. And then at that point, an invite is sent from the system for upload. And the PI or the PI's designee is invited to upload to the system. And it could be multiple submitters. In some cases, the phenotype data come from one institute, and the molecular data comes from a genotype center or a sequencing center. And so that's the point where my team gets involved, and then the data start coming in for a study, and then we start to process the data, and we work with the PIs in an iterative fashion to make sure the data is suitable for third-party distribution. And my team would assign we load the database to or the data to a database and assign sessions, and we create a preview site for the PI to review. And once they've approved that, then we bundle up the data and release it for third-party access and public display for the summary data. As I mentioned, the incoming data flow often, although not always, comes from two different groups. One where it's kind of the cohort or principal investigator, or the sample repository that knows about people, and then a sequencing center or a genotype center that may submit the molecular data. Regardless, the first step is to register the study, and then we want to make sure that we understand the study subjects or study participants and their IDs and any samples from those participants, and the consent categories for those subjects. And those are submitted, and then the phenotype data can be submitted, and kind of at the same time, the sequencing center could start uploading data. We stage and QC the data, and then do the accession and release. When you're submitting, just to give you a really 1,000-foot view of what we ask for, there's some core tables in the submission for dbGAP, and one is the subject listing, and it's very simple. It's a list of de-identified subject IDs that link to each participant in the study, and their consent group. And the consent group is this concept of how the data can be used by third parties. So a consent group might be for nonprofit use only, or for cancer research only, for example. And so this master listing describes how the data from that individual can be used later. And then there's a subject sample mapping table that's a core file that maps participants in the study to molecular samples that they have. And you can have any number of samples per person. A common example of where there's multiple samples for a person is when they draw a tumor and a germline sample from a single person. Well, there's other examples where there may be controls within the study that are sampled multiple times, or longitudinal data where you collect samples over multiple visits. And then so this table is important. This actually links the phenotypes back to the molecular data. And then a sample attributes file. This is essentially any attribute of the sample. This could be where it was collected from in the body, peripheral blood, or it could be a skin sample, but it could actually this file could contain any attribute of how the sample was processed, like the DNA extraction method, or even where it is on the plate when it was used in the study. So we have this kind of sample level attributes file, and then the pedigree information. This is required when you have study participants that are related within the study, and so the pedigree defines the families and the relationships of the members of those families. The phenotype data, which is in a lot of ways the more difficult type of data that dbGAP has to process, is modeled as rectangular tables where the rows are people and the columns are exposure variables or clinical variables on that person. And the submitter needs to compile some study meta information, the core files that I mentioned, and the phenotype data files. Once they've done that, then they can transfer the files using the submission portal, which is an online system where file uploading system, and the data come in through protected servers at NCBI with very limited access by my team and a few other administrators here at NCBI. And then at that point, the dbGAP curators are assigned and they process the uploaded data. And if there's any questions about their processing of that data, they would contact the submitter for clarification. And then the dbGAP staff does these QC checks and loads it to the database, and then we send the variable summaries back to the PI for verification. And for the next couple slides, I just wanted to show you example of incoming data. These are very simplified examples, but it'll give you a sense of what it would look like if you were submitting. So in this case, this is a subject consent file where the subjects are listed here, and then their consent code is listed here. The codes are 1 and 0. 0 has a special meaning that means that they're fully public. 1 in this case represents general research use. So along with the data tables, we ask for a data dictionary. And in the data table, the subjects are rows and variables are columns. And in the data dictionary, each row represents one of the columns in the data set. So you can see subject ID is here and consent is here. Now, when there's coded values, then those would be listed in the values column within the data dictionary. So hopefully that's clear. Here's an example of the phenotype data file and data dictionary. And so this is a little more complex where the codes are more elaborate, but it's the same idea. So it's very simply, the data model is very simple in dbGAP, but it allows a study to be accurately represented without any constraints by us on how the data is formatted. They can just format the data that they have. The molecular data submission, dbGAP really takes any type of omics data, but largely the studies in dbGAP contain either genotypes from arrays or genotypes derived from sequencing data or sequencing data itself, or both. And dbGAP will take things like the raw files that come off the genotype typing machines, like the IDAT files, the cell files. We also take formats like Plink and VCF. These are common formats that are used. Also, vendor software would be formats or processed by dbGAP team. The next-gen sequencing is submitted directly to Sequence Read Archive at NCBI, but so that it would be tied back to the dbGAP for controlled access. The QC checks that we do on the molecular data is we check back with consistency to the phenotype submission. So one of the checks that we do that sometimes fails is actually the gender check. We look at the genotypes to make sure that in the genomic molecular data can be checked back to gender, and we can see if it looks like these genotypes were from a male or a female, and if it looks to not match what's in the phenotype, then we would iterate with the submitter and try to resolve that before it's released. We look for cryptic duplicates. So if you tell us that these samples are from this person and are from two different people, but when we look at the genetics, it looks like they're the same person, then we resolve it to maybe a twin or something like that. But the cryptic duplicates do show up quite a bit in the studies that we're processing. So we look for that, and we notify the submitter when that happens. We do an inheritance check. We check back to the family members because we have the genomic data. We can compare and make sure that people that are listed as being related to each other look like they're in their genetics also related to each other. And then if there's multiple assays on the same person, we make sure that those genotypes seem to be matching. This avoids, remarkably, a large number of incoming data seem to have some errors on when we just do this very simple checking. There's a large number of reasons why that might happen, but things like sample mix-ups happen all the time. And when we're talking about the numbers that are in dbGAP, you would expect it to show up now and then. But since dbGAP's a curated database, we want to make sure that the outgoing data is quite clean. And this is partially because my team just wouldn't have the resources to answer a lot of questions months later or years later after the data is released. So we try to ask all the questions ahead of time. When we're processing the phenotype data, the checking that we're doing is we're just making sure that if you tell us that it should be based on the description or the data type in the data dictionary, that this should be an integer, and we see some text word, then we'll ask you what that is. We want to make sure any encoded values are described, and so that the end user would know what number 2 means in a code. We make sure that the description is understandable at a basic level, and then if there's things like height or weight, we know what the units of those are, so that there's no confusion later. The core tables that I described earlier are checked back to the phenotypes files for consistency. For example, if you have in your phenotype file this person is a female, but in the pedigree file it's showing that person as a father of that family, then that would fail our check. Remarkably, we do see problems like this in the data we're processing, but we try to resolve all those before they're released. We also look for things like addresses and names showing up in the data, and we would ask the submitter to remove those before we would release it. We also process, as I mentioned, these study documents, and these could be protocols or data collection instruments, questionnaires, that type of thing. And our processing is that we will actually annotate documents back to the variables, the data, and we can accept pre-annotation by the submitter, or we can do it ourselves in some cases. And we do take the documents and mark them up in XML, and we either do that in-house or by vendors, but this markup allows us to display on the public side links from variables to the questions or how they were collected. And then once you have configured your submission, I just wanted to show here the submission portal is used. So I'm just going back to this original flow chart on the submission, and where the PI is invited. On that invite, there's kind of a 1-week acceptance period. But once you accept the invite, that access doesn't expire. You can submit to that study at any time in the future. Here's the email that goes out, and then this is what the interface looks like to upload files. It's very simple. You can add submitters if you're a manager. So for some studies, it requires multiple people to actually upload. But essentially, what you do is you just choose the various core files and upload those, and then down below you can upload multiple phenotype or genotype files through the interface, and then you press submit, and that's it. And the history is tracked. So the submission itself is not so hard. What's hard in dbGAP is formatting the data to be submitted and the registration step. So now I think we're getting towards it's about 35 after. So I did want to show a bit of how dbGAP looks on the public side, and how to search for data. So when you come to the dbGAP homepage, you see here there's a number of links. The top one here is advanced search, and that would allow you to search for data of interest if you were interested in maybe obtaining data that's available in dbGAP. And I'm just going to go to a live demo of that. And when you open up the advanced searches, the way it works is it's solar faceted. So this is similar to how Amazon and some other popular websites do things. Essentially what we've done is we've taken all of the data in dbGAP and indexed it back to different categories. And these categories we call facets. And then the values within the facet are selectable. So at Amazon, this might be electronics, and then these values might be TVs and VCRs and things like that. But in our system, we have facets that are disease focused. So you could look for diseases that might be of interest to you. And if you wanted to search for for example, bipolar disorder, you could choose that, and then you'd see that there's 3 studies in dbGAP on bipolar disorder. And then you could you would also see that there's about 86 or 8,600 variables for those across those studies, and then the phenotype data sets, any documents, and then molecular data sets. And so this is just a public description of how those look. When you look into the variables, we have variable pages that show, and in this case, I just took a family genomics of bipolar disorder as a study, and they had a variable description called body site, and this is a very simple variable, but you can see that body site is actually it just shows the distribution of in this case the case control status for bipolar disorder. The various facets can be combined. So if you were interested in studies that are from, hold on, there we go. So I'm just clearing out these facets. So starting from the, you could choose that you want studies that have whole genome or whole exome sequencing and a specific disease, and the combination of the facets would limit you down to maybe one study that meets that criteria, and so on. And you can click off the facets here. So hopefully that would give you this shows you how you can search in dbGAP for various data that might be of interest for your research. Now I'm just going to close that. I just wanted to introduce the advanced search there. And then now that we're getting towards the end of this, I wanted to show just briefly where you would go if you actually were interested in requesting data. So that's a separate interface. The public interface just shows the summary, but to actually request the data, you have to go through a controlled access system. And that's the second link here on the dbGAP homepage. And as you click on that, you'll go to an interface to access. But I wanted to kind of step back a bit and say, before you go to request data, there's some information you should know. If you're an NIH intramural researcher, you actually have a permission form to establish essentially the signing official that will sign in your application for dbGAP. All other researchers that are external to NIH would have to have an ERA account. Now, many U.S. researchers have ERA accounts because this is the same account that you would have for an NIH grant. But if you don't have one, it's okay. You can get one. But this does require your institute to actually get an ERA account. And so you can follow this flow chart and hopefully get your account set up. And once you do, then you can start to log on to dbGAP with authorized access. Now, if you already have an ERA account, you can kind of just flow right down to this first step. Once you do the application, it's an online TurboTax type application, and you complete it, the application has to go to one of your signing officials, and this is completed within the application itself. You choose a signing official. Then the signing official signs the application, and then that goes to the dbGAP data access committee for that data set, those data sets. And if it's approved, then you will be sent an email on how to download the data. And if it's rejected, then you may have to revise the application and go back through this loop. The last thing I wanted to tell you is this step here. There is actually a YouTube video for this. So if you just go to YouTube and type in dbGAP, apply for dbGAP access or dbGAP authorized access, you can find that video and see a 3-minute video of how to actually do that process. With that, I think we're at about 1:45, so I will open it up for any questions that anybody has.
Great. Well, first of all, thank you very much, Mr. Feolo, for a great webinar. We did get a question in the private chat that was sent to me. It's a little long, so I'll read it out for you.
Okay.
So with the development of large-scale genomic resources or clinical data, including the mouse genome initiatives, where do the thousands of labs fit? What I mean is, can you speak to the integration of the large-scale data where a relatively complete phenotyping is achieved across thousands of gene knockouts, as we see with the COMP/IMPC, maybe that's COMP/IMPC, and 23andMe or Genomics England efforts, and how the individual investigator can best contribute his or her so-called long-tail data to this emerging world? If you need me to repeat any section of that, please let me know.
Well, I think I can't address maybe the main issue, but maybe related to that question is dbGAP is primarily used for human data, so we wouldn't have any mouse data, but we try to integrate the best we can, although it turns out that if you've seen one dbGAP study, you've seen one dbGAP study. They're all different. There are exceptions where there's large projects that are NIH funded and they try to coordinate things like phenotypes and how the molecular data is processed and things like that. Although it's not to the level that one would wish, but I don't know if there is a good solution at the NIH, at least for that at the moment, but I know people are working on it.
Excellent. As far as our questions are concerned, we did have one other, but it was in regards to the slides, which we actually covered during the webinar, and I responded to them privately in regards to it as well. Mr. Feolo, is there anything else you'd like to add at this point?
I think, you know, I have one last slide, and I just wanted to thank my team members, and they're listed here, and this dbGAP actually happens with many other people at the NCBI, and wouldn't be available without the support of the NIH, all the data access committees and genomic program administrators. But especially it wouldn't happen without the submitting PIs and their staff. So I wanted to thank them also.
Excellent. Well, Mr. Feolo, thank you once again for joining us. And I'd just like to take a moment to put in a plug for our next webinar. We will be taking a break for a few Fridays throughout the holidays, but our next anticipated webinar will be on December 8th, Friday at 11 a.m. Pacific Standard Time. And that will be on hypothesis with Dr. Marianne Martone. Once again, thank you to Mr. Feolo for joining us today, and also thank you to all of our live participants, and thank you to those watching on YouTube. This recording will be on YouTube in a few hours. I'm going to paste where this will be accessible. It'll be accessible in our newinfo.org/about/upcoming webinars tab, where you can see our upcoming webinars and also our past webinars with those YouTube links applied. At this point, I think I'm going to end the webinar. Mr. Feolo, does that work for you?
Yes, thank you. Thank you, Devin.
Absolutely. Thank you so much.